My first slide actually is just going to be a photograph of Susan. And for those of you that didn't know her, she was a true force of nature. And we miss her daily in a whole variety of different ways. And we were in this room not so long ago remembering her. Many of you might have been in here as well at that time. And everything she did for this community. The fact that we're all here today, the work I'm going to talk about, was all possible because of everything that she made happen. And so I just wanted to start by acknowledging that. The other important thing I must acknowledge at the start of this is that this work is, I stand here and I get to talk about it, but it's really because of all of these people. It's a pretty big team that work on the platform I'm going to talk about. Everything from engineers to data scientists, biologists, and we make very hard work sometimes seem easier than it should be, and it still takes an enormous amount of work. Even with all the robots I'm about to show you, there's an enormous amount of effort that goes into making a stem cell line, differentiating it. Even with robots, the work doesn't get easier. In fact, maybe it gets harder because we just anticipate that we should be able to do more and more. And so a huge shout out to that team. So we have a view that discovering new therapies, new treatments will need aspects of scientific convergence, and so part of that is needing things like robotic infrastructure to be able to do this. We need higher throughput assays and things that are both disease-specific but also quite agnostic at times, so that you can go into things in a little bit of an unbiased way. And then once you're able to do that with having large amounts of data and disparate different data sets, you need then the capabilities to be able to analyze it in various different ways. And so at NYSCF, we have done this in a really concerted effort by bringing together different groups from different backgrounds. And by that I mean we have hardware engineers that have put together robotics, developed underlying infrastructure, do a lot of process development to get things operational. A whole then raft of computer scientists and data scientists that have built things from LIMS systems to running the analyses on what we do. And then obviously the biologists as well. And so from a whole different set of backgrounds, from gene editing, differentiation, disease-specific backgrounds, bringing all of those together, and it's at the intersection of this that everything I'm about to talk about can happen. And one of the big words that we talk about fairly often is scale. And I think for many different people in this room, scale might mean something different. If you're in a therapy space, it might mean billions of cells. Maybe in a screening space, it might mean billions of cells as well. But one of the ways we think about scale is outscaling in terms of the number of cell lines that you can analyze in any given experiment. And so this concept of scale is something that we think about a lot. And so to get to that point of scale, whether it's reprogramming lots of lines in parallel, differentiating lines in parallel, you can obviously throw lots of hands at it, and you'll get all sorts of technical noise from people just doing things differently, having had coffee in the morning or not had coffee in the afternoon, all sorts of different things. We turn to robotics to be able to help us overcome many of these aspects. And so in the lab, there's a whole series of different clusters of instruments that move plates around, feed, transfect, passage, thaw, freeze, you name it. As a biologist, if you're in a tissue culture hood, these robots can basically do much of what you might do by hand, and obviously in a fairly scaled way. There's centrifuges, imagers, incubators, you name it. It's all sort of hooked into the robotics. And in terms of that word of scale that I was coming back, no day is typical, so that's kind of a bad title here. But a typical day for something that we might be dealing with might mean that we have, tens of thousands of wells growing different kinds of cells. It might come from hundreds of different people all growing in parallel in different areas. That might mean that with clones and differentiations going on, there's sort of thousands of cell lines individually growing. And then everything has to get imaged. And then all that data has to get crunched and put into the database and models that run off of it. So an enormous amount of work, as I said, that goes into what we do. But underlying much of this is really developing platform technologies. That's really what we're sort of focused on. And so when we started building the robotics, it was really with this in mind, taking large cohorts of individuals, reprogramming them, whether it was skin or blood, putting them into plates and allowing you to expand them. And that was what we did sort of back in 2015 and sort of the backbone of a lot of the work that we've gone on to do. It's allowed us to build things like a repository. If you're looking for cell lines, you can certainly come and get them from the repository. There's a lot of different diseases, ages, ethnicities, all sorts of things buried in here, tied to different data sets as well. But along the way, we also build different tools that we've needed in-house to be able to solve different problems. If any of you do single-cell cloning, trying to figure out whether it came from a single cell at the beginning can be an arduous process, and so a byproduct of having automated images means we can build models that allow us to detect whether there truly were single cells. But we also generate a lot of plates, and we need to know that they're in focus. And so having very rapid tools for looking at focus assessment is very important. So we, on top of it, have a lot of differentiations. So we do this in various different ways. And as I said, it's really all steps of a process that might go from thawing to passaging, feeding, transfecting, all baked into what the robot can do. And that lets you ultimately then create plates and dishes with neurons that look like this. And we've used these in a whole variety of different studies, some in collaborative things with people in the audience. And one of those, it was sort of touched on a little bit last year by Kristen Brennan, but we were studying or trying to model PTSD in the dish. And so for this, we took combat-exposed veterans and turned them into multiple different neuronal types using NGN2 or ASIL1 and DLX. And looking at the response that you saw following the exposure then to a stimulant. And so in this case, it was hydrocortisone, and we saw that there was a PTSD-specific expression signature in that cohort. And so the data's now being further refined, further analyzed. There's additional data that's come on top of this to answer the question of why do PTSD cases hyper-respond to hydrocortisone? What is this driving this difference? And so, it gets you to this point where you can start sort of thinking into, are things genetically driven? What cell types does this act through? We can expand the number of cells that we can look at, what mechanisms, and then ultimately, you can validate it in vitro. So tying together, clinical data to in vitro data. And a great example of that, and we'll hopefully hear more about it on the panel, is sort of in the INDIE program that Dan will talk about, where taking a similar approach of modifying many genes, running large-scale differentiations, and then looking at the data sets that come out of it. This is work that's led by the CARD Center, and again, another collaborator of ours where we've been making cell lines with them. I think ultimately, though, we want to end up getting toward this sort of stage where you have large numbers of cell lines, you're able to use them perhaps with automation, but ultimately to get to a point where you're screening drugs and screening therapies that could ultimately be used to treat diseases. But the gap between that is actually quite big, and so to do those things you need assays like cell painting that we'll hear more about and touch on in a second, I'm sure. Imaging-based could be transcriptomic-based, whatever it might be, to get to the point that you can actually then do the screen at the end. And so for us, one way we've approached this is taking large cohorts of samples. So here it's skin cells. These are fibroblasts from a fairly large cohort in here. Each square is a different person. And just looking at it, you can see density differences maybe, but it's really hard to actually tell the difference between what's going on in any given cell here, in any given well. And so there are tools that allow you to dive in, and Beth will talk about this with things like Cell Profiler. But for us, we switched to tools, things like deep learning as well, where you can begin to do this. And so last year we published a Parkinson's study where we basically said, if I take enough, oh no, I pressed the wrong button. There we go. Okay, if you take a large enough cohort, can you derive something called embeddings from the images and classify whether another image that they might look at is from a healthy person or a diseased person? So we did this in Parkinson's disease, used the robots to grow lots and lots of cells, ran cell painting many times, and then imaged and put them into a model. One surprising thing that comes out of this is when you just run basic clustering on the embeddings, is that things group together. And the reason they group together is that there's very interesting signatures and fingerprinting that come straight out of the cells, which is pretty remarkable in and of itself. So something just to bear in mind is just from images we can tell sort of who is who. We've done this, so this was the results from Parkinson's, and really the thing you want to be focusing on is how far to the right it's going. That's sort of a measure of how good the model is working. So the further to the right is better, 0.5 would be random chance. And so going through this, when we get to specific disease populations, we have very strong ability to detect this. which is incredible considering this is just coming from skin cells. And so a question that we can then tell the difference. But a question that we get a lot is, but what is it seeing and what is driving the difference? I'm going to click through really quick. And so we've built tools as well. Like I said, that's one of the main things that we do. And so things like scale effects allow us to dive into this, similar to sort of how Cell Profiler works, but amenable to these very, very large screens. So this is sort of a pipeline that we've really been working on, and we'll hear from others on the panel in a second, because it's sort of the same approaches that Recursion use, and we'll hear about that in a moment, and similar things in the JUMP data sets. I'm going to be one more minute. I apologize for running over. Other questions that we've asked are things like predicting aging. Can we predict the age of donor samples at the time they were collected? And if we refine the question, it's maybe, can we predict middle age? So again, looking at cells in the dish, can we make a prediction about what biological age they are or were? And so if you train went too fast. If you train a model on young and old samples, these ones here in the middle are the predictions for sort of middle age, and it corresponds remarkably well. So even just, again, looking at these skin cells, we can sort of get an indication of biological age from these data sets. And so the last one I'll just touch on is a rare disease that we've been working with called infantile neuroaxonal dystrophy. It's a very, very rare disease, ultra rare, but causes a number of different symptoms, and sadly the children affected by this don't live much beyond 10 to 12 years old. And so we've been taking, again, cohorts that we've collected in collaboration with research foundations and developing disease-specific models using these same approaches of cell painting and screening to develop models that are very robust, allowing you to separate out between clusters. If you were to cluster these, again, you can see that the disease cluster separates from the healthy. And the question we obviously want to get to is, can we revert this? Can you make these sick cells appear healthier again? And so we've been screening things like the FDA-approved library, looking for this. And what comes out of it is that you can find compounds that do indeed show a dose-response effect. And so the higher here they go, the healthier they are getting. And so this is quite exciting, especially as some of these hits now are converging on pathways that really do make sense for this disease. So apologies for going over a little bit, but I hope that introduces sort of what we're going to hopefully talk about on the panel now. And yeah, again, thank you to all the people that did this work, because it's really thanks to them.
Thank you, Dan, for that really quite comprehensive slide deck where there's a lot to show. It just shows the scale of what you do at the NYSCF. And so to tee it up, we're talking a lot about science and how stem cell technologies can help us understand diseases. But what we're also hoping is that this will eventually lead to new therapeutics. And I won't say how many years, because it's been many decades that I've been doing this, but the biggest issue we have in drug discovery is really choosing the right target for the disease, and that you'll see efficacy in the clinical trials. Most drugs will fail in a phase 2b trial because of lack of efficacy. And part of it is because maybe we didn't choose the right target for the disease, or perhaps we didn't have the right biomarker to be able to determine whether the drug was actually having an effect on modulating the target, or maybe didn't quite select the right patients for this disease. So there's a lot of issues, and hopefully we can get the panel's perspective on how we can change that and accelerate drug discovery. So please come on up. So first I'd like to introduce our panel. So I introduced Dan. Next is Tim Ahfeldt. He's currently at Recursion, and he trained with Dr. Chad Cowan's lab at Harvard for his PhD. Then he did his postdoc with Lee Rubin, where he carried out translational research using stem cell models of Parkinson's disease. In 2017, he joined Mount Sinai as assistant professor in the Neuroscience and Neurology Department. He is now VP of Advanced Translational Modeling at Recursion. Tim is shaping the neuroscience scientific strategy and spearheading the expansion of the Recursion OS platform to include multimodal maps of the human brain. So the long-term goal at Recursion and for Tim is to identify new therapeutics for patients suffering from neurodegenerative disorders. And most interestingly, if you've read his bio, is that he has an interesting past. He worked as a police detective in Berlin before starting science. So, so detective work will then help us with neurodegenerative disease. Next, we have Beth. Dr. Beth Cimini is senior group leader and CZI imaging scientist and head of the Cimini lab for imaging platform at the Broad Institute in Cambridge. She obtained her PhD in biochemistry and molecular biology with Dr. Elizabeth Blackburn at UCSF, studying variants of telomere master scaffolding protein TIN2. She then joined Dr. Ann Carpenter's lab at the Broad as a postdoc and then as a staff scientist to focus on cell imaging analysis. Her lab and the rest of the team have created the bioimage analysis tool called Cimini and maintains the cell profiler as well as applies open source tools to novel biological problems. She is among many other affiliations, but most importantly, she's the president of the Society of Biomolecular Imaging and Informatics, or SBI2. And last but not least, Dr. Daniel Ramos, who's joining us from National Institute on Aging. Daniel is a scientist in the iPSC Neurodegenerative Disease Initiative, which you just presented, the INDI, at the new Center for Alzheimer's and Related Dementias, which is CARD for short, at the NIH. He obtained his PhD with Dr. Charlotte Sumner's lab at Johns Hopkins, where he studied infant neuromuscular disease, spinal muscular atrophy. He explored the human biochemical response to the first FDA-approved treatment for SMA using patient autopsy samples. His current research at INDI focuses on understanding the fundamental mechanisms of dementia through use of isogenic iPSC-derived neurons. So we'll start with Tim because from the drug discovery aspect, you're at Recursion, and I thought perhaps maybe for those of you who don't really know Recursion, you can give a little bit of an introduction to this company and what your aspirations are.
Absolutely. Is this on? Test. Oh, yeah. All right. So, yeah, Recursion is a tech bio company that I joined about 2 years ago. And some people ask me why tech bio, not biotech. And the difference is really, who runs the show, and and also what do we emphasize, right? And so at Recursion, we have a strong belief in the power of AI, nowadays foundational models, to understand biology in a way we are calling this is we want to decode biology to radically improve lives. And this AI approach is supposed to touch every aspect of the discovery all the way to the clinic. And so when you think about this normal V-shaped funnel that we always see in every drug discovery talk, we want to turn this into a T. We want to broaden the actual funnel. We want to fail faster. And this broadening would be, with technologies very similar to the ones that Dan P. introduced today. And then we want to narrow the neck of the funnel very, very quickly. So we want to fail faster using AI approaches. We want to get to better target deconvolution using AI approaches, in particular digital chemistry. And so accelerate down there and make a more ideal pipeline that is maybe more efficient than the one that we have today. The work that I'm bringing to Recursion, or let's say a lot had been built before I joined Recursion. This included, these AI approaches to understanding biology, understanding phenomics observations. And they had installed an incredible automation approach, a scaled semi-automated lab. You introduced aspects of this. And what they didn't have were advanced in vitro culture models to actually understand the biology. And I think in this room in particular, I don't have to convince anybody that there is a, kind of a case that we can make for the intersection of a perturbation, genetic or chemical perturbation, with a specific cell type environment that leads to a relevant pathological phenotype, only in that environment. And so this is the new thing that I was asked to lead at Recursion, particularly in neurosciences, together with our partners Roche and Genentech, who are working together within the space of generating neuronal maps of biology and then identify new therapeutic avenues really far away from the beaten tracks that we normally, with our hypothesis-driven research, step on. So that's the synopsis.
Right. Large scale. I mean, if you go to the Recursion website, they generate 2 million data points per week. So, talk about scale. And I guess AI and machine learning is where it becomes important. But also importantly, from all this imaging data, and Beth, you've been maintaining and working in cell painting and cell profiler. Maybe also, just for those of you who don't quite know what cell profiler is, maybe you give a little bit of a description, and how and as well, as you think about, as you talk about cell profiler, your software, I also want you to also talk about the fact that you're able, using this technology. You just recently, she co-authored a paper where they were showing the power of combining genomics with high-content imaging in iPSCs sourced from hundreds of unique donors. And the results showed associations between rare gene variants and cellular phenotypes. So it's a very powerful technology here. And so, Beth, if you want to describe a little bit about your work and how this is enabling.
Yeah. Hi, folks. So I'm part of the imaging platform at the Broad, and yeah, we design software and assays. That's where the cell painting assay, which we mentioned a couple times, which I'm going to assume not everybody has heard of it. So the idea is essentially, rather than tagging your favorite protein or your favorite marker, what if we just added organelle dyes? And we just looked at where the organelles are, and then we just measure many, 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 many features, usually about 8,000 features of each cell. And as someone who was trained in molecular and cell biology, when I joined the group, I was like, there's no way you learn anything from that. That's silly. Like, why? But it turns out that you can actually learn quite a bit from just deep measurement of organelle dyes. And the way we do that deep measurement is a software tool called Cell Profiler, which is an image analysis workflow tool. Possibly some of you in the audience know how to code. If you want to learn how to code, coding is great. Coding shouldn't be a requirement for doing good biology, in our opinion, and so Cell Profiler is an image analysis workflow tool designed to allow you in a no-code way to develop workflows, whether you have one image or several million images, and we use it at both of those scales. And so being able to then sort of say we're going to measure 8,000 features in each cell, we can actually find, as Christine just pointed out, connections between how cells look, even if we're talking about iPSCs for a neuronal phenotype, or just sort of, again, like looking for rare SNPs and rare cell features. We can actually say with organelle dyes in an assay that is about between $1 and $2 per sample once you're at scale. We can tell a lot about what pathways are active in these cells or what pathways are deficient in these cells in ways that I totally didn't believe when I first started doing it, but it absolutely does work. And so getting to do those kinds of approaches and getting to build these databases like the JUMP Consortium, where we've got now tens of thousands of perturbed genes and hundreds of thousands of compounds, that data is all freely available to everybody, so have fun, has been an incredibly fun last few years.
So on that, though, it generates so much data. How do you see the importance of data standardization or assay harmonization as helping stem cell research?
Oh my gosh, it is 90% of the battle. So again, I'll briefly mention this JUMP consortium, which was touched on. The idea was, what if, we had someone who was leading the group who was an actual wizard, Ann Carpenter, and she got 10 different pharma companies, as well as a number of nonprofits and universities, to all work together to make one public data set. Again, I don't know how she does it. But physical compound plates were sent in the mail between these 10 different pharma companies. And now we have data where we have the exact same compounds, the exact same layouts in different physical locations. And not every company had the same microscope. So there's a great new preprint. It's Arevalo, Arevalo et al. On bioRxiv, where John was now able to take this data that we now have and look at how well can we batch correct the exact same cell culture, the same vial of cells from ATCC, the same batch of reagents, and how well can we correct within an individual lab across days? How well can we correct across microscopes that are in different facilities? Or somebody who has, somebody has the molecular devices IXM, somebody has an Opera Phoenix, and every level of that, you get batch-to-batch variability. And every level of that, it gets harder to correct out post facto. So the more of it you can correct up front and the more you can standardize up front, the more likely that after all your correction, your biology is going to be still sitting there at the end of the experiment.
Great. So Dan, you presented, the scale, and you deal with large data sets. What's really interesting is that the NYSCF technology and all of the labs that you work with and collaborate with, you do have a really nice success story in that the team discovered how to repurpose a drug for ALS using this platform. And it sounds like, Beth, what you were just saying, how it's important to standardize. You generate so much data. How challenging is it to uncover these phenotypes from the noise? There's subtle changes.
Yeah, what a great question. And something that I talk about with the data science team day in, day out. I think every day we have some kind of conversation about metadata or extracting the right information from a data set. I think we were chatting about this before as well. I think there's an element of more is more sometimes, and if you have more, you will enhance getting out of it a phenotype that you might be looking for. And if there are aberrant signals in there that are just noise and are artifacts that are coming from this, they'll get drowned out by the good data that you do have because the data sets get so large. And I think that's like a really important thing of why this is hard to do. I appreciate that, a bunch of us sit here with robots in our labs and great computational power and all sorts of things that isn't available to everyone. But I think that's where, fundamentally making data sets available, like both Recursion and the Broad have done, gives then training data sets that can be used by anybody here in theory to go off and run your own experiments and already have a lot of data to fall back onto that's come from another model. And so again, helps maybe add more power to your experiments as you go forward. Doing things, we spend a lot of time thinking about plate layouts, like where do you put samples in plates and from plate to plate and batch to batch and moving things around, because again, these analysis approaches are very sensitive. They'll learn patterns very quickly. So if you always put your cell in A1 of a well, it will just learn to expect that it's going to be in there, or you always put the same drug in the same well, it will learn that those things are just, they correlate together and will lead you down the wrong path. So all these things that we think about in designing an experiment hopefully weigh in then to any signal you are detecting at the end of it is a truer signal and not just a byproduct of noise of the experiment.
Great. And Dan Ramos, we have two Dans on the panel. Your work focuses on robust differentiation protocols for neurons and using CRISPR engineering to generate highly characterized iPSC models. Why is this so important, and how do you think this will impact research in neurodegenerative diseases?
Yeah, thanks so much for having me. I really appreciate being here today. I just as a primer, Dan to Dan. And I gave a primer on Indy, but for folks who may not be as familiar with it, it is a relatively recent project started at the NIH in the Center for Alzheimer's and Related Dementias, CARD. The overarching goal there in Indy, the iPSC Neurodegenerative Disease Initiative, I don't know if I did define that for you, but is really to try and understand what are the fundamental, essentially disease molecular pathways that are set off by in the course of disease, in particular in Alzheimer's and other related dementias. So the scope of it for us is modeling over 100 different familial Alzheimer's and related dementia mutations in highly characterized iPS cell models. We do really rigorous sort of characterization of iPS cell lines to just make sure that they're, I'm sure everyone here who works with stem cells is very familiar with each stem cell line you work with is going to behave completely differently. Some grow a little bit better, some are less, some are more sensitive to spontaneous differentiation, those sorts of things. Like we put our iPS cells through a rigorous testing panel. And really the goal there is, there's sort of two phases of Indy. The first is this is the genome engineering portion, which we're making heterozygous, homozygous mutants, versions of each of these variants that we're interested in, and also halo-tagged wild-type and mutant proteins for each of those proteins of interest. So we're really trying to get a comprehensive set of iPS tools, and importantly, when we're also engineering these iPS cells, and this tries to get back to the noise issue that comes up all the time in experiments, is in addition to making those mutations, we're actually also using, and we use CRISPR to introduce all of these mutations. We also revert them back to the sort of wild-type state, really to try and control for any potential off-target effects. CRISPR's excellent, it's not perfect. So this is a way that we're going to build in an extra layer of controls to our experiments. All these cell lines are going to be made available, whoever wants. Have I been doing that the whole time? Okay. Let me start from the beginning. So, we partner with JAX-NYSCF Laboratory. They're, typically the mouse people. Now they're also getting into the iPS cells, and so the repository is going to be online, whoever wants to use them. In-house, we also do we're starting to do deep characterizations of what are the effects of these mutations on downstream assays, proteomics, transcriptomics, microscopy-based readouts, and in my main wheelhouse is actually in the context of CRISPR-I and CRISPR-A screens. So in the context of each of these mutations, how does that interact with other genes in the genome? So we do genome-wide, our ultimate goal is genome-wide CRISPR-I and CRISPR-A screens in order to understand really the core biology of these mutations and how they may genetically interact with other genes in the genome. It's a way to really try and understand the core biology of those disease mutations, and also opens up the door for new target discovery. So in that vein, I mean, some of those things that, why is this really important to us? So we definitely, in Indy, we're also trying to implement some of these similar strategies where automated cell culture, we are sort of, I feel like every time we run into a problem, it seems like NYSCF has encountered that as well and has solved it. Or not solved, but progressed and advanced. So, it's always you're moving the needle. And so we're actually, running into, we're setting up our automated cell culture system, trying to figure out how best to run these experiments. And so we're starting with a very reductionist approach, where we're taking a very well-trodden differentiation protocol, the doxycycline-inducible neurogenin protocol, differentiating our iPS cells into these cortical-like neurons, and then subjecting them to these downstream assays. And it is reductionist, but it's one of the strategies that we're trying to employ, just to not necessarily say that we're going to understand how to cure a disease after we run our experiments, but really to just try to understand some of the basic biology that may be, just in the context of these disease mutations. And we're also, in microscopy areas, we're also trying to understand how best to run these experiments. How can we reduce complexity, at least for our experiments, and trying to move more towards pooled culturing protocols, where you're not just separating mutant A over here and mutant B over here on your plate, but there's actually really cool technologies nowadays. Single cell is one of them that just will allow you to basically pool multiple cell lines and then demultiplex that at the end of your experiment. And there are new tools that are going to let us do that optically. So I guess for that, it's really trying to just understand the core mechanics at play in the disease, reducing noise, reducing complexity of experiments. So yeah, that's sort of our approach. But yeah.
I'm so happy to hear how the effort you're putting into this work, and in terms of standardization or trying to harmonize ways of running an assay, how you plate the cells, where you put the compounds in the cells. It shows the amount of rigor that is required, and it's not typically something, I'm from industry, and it's not something that we've typically heard from academia. So it's wonderful to hear, this amount of effort on that. And Tim, you have experience both in industry and academia. And, what's your perspective on how to accelerate the knowledge transfer from academia to the industry?
Nice, yeah. It's a topic. Is it on again? Still not working? Test. Yeah, good. Okay. It's a topic that's very, very important to us at Recursion. We thought about this as well, because this cross-fertilization goes both ways. It's beneficial for everybody. And we heard already about the release of JAMCP as a data set. You had mentioned our release. We have just a couple months ago released a relatively big data set, 2.2 million images. So that represents about a week of full-speed screening for us. And this is available for any academic to use, and it's really powerful. It comes also with a already pre-trained deep learning model, relatively clear instructions on how to leverage this. And so anybody could explore a phenomics data set that spans almost the entire genome, about 17,000 individual knockout perturbations in this data set, and a number of pharmacological substances that we put into the screen with very diverse functions, well annotated functions. And so you can really begin to understand, what we are calling a shared embedding space in a phenomics model approach, where you can understand kind of the converging biology or the systems biology. You can see, your ribosomes falling together with your ribosomes, your proteasomes with your proteasomes, but you can also start to understand, make new connections within there. So you can rediscover some of the I want to say, canonical biology of the last decade in this data set, and you can begin looking into the areas that you find interesting, and then put this in context with, a compound, an inhibitor, or potentially an activator. And so we love everybody to adapt this. We would love to get feedback from anybody here in this conference on the data set. Reach out to me if you want to work with this data or you make any progress on it. It would be great.
Great. One of the aspirations, I guess, of stem cell technologies and all of the technologies that you're all enabling is potentially the one day to be able to have a clinical trial in a dish or avoiding animal models altogether. One of the issues also with drug discovery is not just whether you have efficacy in the clinic, but we also have tox. And Dan, just wondering what your thoughts are. Do you think we'll be able to predict tox in a dish?
Yeah, it's a great question. I'm going to answer. I just want to add one thing. I think it is so good, all of the open source stuff that is coming out of this space at the moment, though, because it changed software engineering once everything started being open sourced. And the fact that things like lab notebooks are very expensive if you want to use one of the commercial systems, or if you want to use some statistical software, not to call them out, but Prism is very expensive to buy licenses to. And the fact that more of these open source tools are coming, and then data sets as well on top of this, it really, take advantage of it, because it's incredible. Toxicology, sorry, back to toxicology.
No, go ahead.
Yeah, so I think we're seeing all sorts of things from the stem cell space, being able to make predictions in a whole variety of spaces, whether it's from does one given drug work differently in this individual versus this individual, and what drives that? That's one level, and I think we're certainly seeing that come out in different ways. In terms of toxicology, almost we were a little ahead of ourselves when we tried to embark on this, but a few years ago, we actually embarked down a path of could you replace an assay? So there's an assay that is commonly used in the pharmaceutical industry called the EST assay, and it's where a given compound that's early in the pipeline will be put into, it's a mouse-based model, and they look for whether it perturbs the formation of cardiac tissue very early on. So they take some embryonic stem cells from mouse, group them together into a little embryonic body, and it starts beating within a few days. And so they look for teratogens, the things that will disturb that very early in the compound process, because if it's going to disturb that formation of heart tissue, it can't go into people. Like, it would just not be safe. And so this mouse model exists for this. And so there was a thought of, like, could you humanize this, and could you turn this into more relevant by using human cells? And so even there, we found really encouraging data. It's definitely doable. We were working with early training sets, and we haven't gone back to revisit those data sets, but now we didn't have these tools necessarily quite at our disposal to be able to really dig into this at the time. But you think we were using gene expression, but you think now combining this with imaging, 100%, like, whether it's finding new drugs, finding their toxicity state, finding their efficacy, it's going to change.
Yeah, the one thing I'll just add is the follow-up to JUMP that is now being planned is a thing called OASIS, which I never remember what it stands for, but is the idea of basically, can we do exactly that? And the O is for omics, and can we use cell painting, but a lot of other omics as well, to say, can we predict toxicity, and can we get stuff to fail faster? And so that consortium is being pulled together right now, because we think that probably imaging will be one of the good ones, but that several omics can probably help us get there so that we can fail stuff out faster and easier and cheaper.
I think Tim said the word multimodal. It's, the stitching together of these disparate data sets where you might think of transcriptomics and RNA sequencing and genome sequencing and then imaging. Don't think of them as like isolated experiments. They are things that should ultimately be tied together in singular models, and that will be where more of this gets unlocked.
Can I just add one more thing before then? That I wanna backtrack one thing a little bit. I think these consortiums are very important. I think all of the standardization is important. Being able to scale this stuff is important, but I feel like I also want to make very clear that rigor doesn't mean robots, that you can do these sorts of assays in a standard academic lab. In fact, we're co-authors on a paper that just came out, I think like 2 weeks ago, where a group here at Rockefeller was doing cell painting to look for drug resistance. And all of that data, it was 20-something batches of data, was hand pipetted by fantastically talented graduate students. And they did a beautiful job, and we were able to find what we wanted to find. And so I think these big consortium efforts are important. Big data releases from groups like Recursion and like the consortia are important, but don't make that think that none of this is relevant to you and that this isn't something you can't go back to your lab and do yourself, because if you're careful and thoughtful, you can.
Can I then tag one more? And this is really good, because this is where this is headed, though. Probably some people that roll their eyes. I love a particular robot we have in the lab called the Opentrons, because it's designed to make it accessible to like just any lab. These are $5,000 to $10,000 robots. That's not an outrageous amount of money, considering that bigger robots are hundreds of thousands of dollars. And they will take then the ability to kind of do these reproducible staining experiments or these simple assays. And so as we see, again, the open sourcing of software and the reduction in price of like this kind of equipment, it opens the world up even further. So yes, by hand, totally, I completely agree, but there's accessible tools coming outside of big labs that have this giant equipment.
So any questions from the audience?
Thanks for the panel session. I'm wondering, so I think it is believed that to process or enhance the kind of drug discovery process, it is believed to that simpler system. as a model for the recapitulating physiological or disease states. But now there are so much advancements in the technologies such as unbiased predictions or automations and every kind of like something that just a hand pipetter kind of scientist could not do in the lab is now possible. So I'm wondering whether your kind of thoughts and insight about whether drug discovery process will be still relying on a simpler model system or more complex kind of model system in next 5 or 10 years.
Anybody on the panel want to take that?
I can take it, but go for it.
All right. So I think we want to right scale our models to the question we have, right? So we have made a lot of progress in the inference space. We don't have to run every experiment anymore. We can, make predictions based on a subset of experiments that have been run. If we believe a phenotype that we really care about, or a number of phenotypes in a disease area, that there are cell autonomous effects that we really care about, then I would suggest, working with a single cell model and have the best signal to noise ratio and the highest chance of discovery. If we think it is a more complex phenotype, we have some understanding of the underlying biology or pathology, and we need these interactions. Joe Blanchard gave a great talk yesterday about this, right? About the need and the interplay of different cell types. Then you want to increase the complexity, and with that, you will increase your operational burden, the operational costs, and probably reduce the scale. So it's a fine line to navigate, and I love that we are now in a space where we get that experience. We have the right computational models to explore complicated models. This wasn't possible just a few years ago. And so, yeah, now it's the time to really see this value proposition mature a bit.
And I'll just add to it. I think there's like the things that we don't always spend a lot of time doing are like the actual very, very basic optimizations of a protocol. So yeah, we can make a few cell types pretty robustly, fairly well now, because the protocols have got very good, but there's a lot of cell types we can't make. And I was going to call Joel out as well, because that's a great example of like, the complex things where you want to put things on a chip, or you want to, these things aren't going to scale yet to these kinds of things. And so, no, there's plenty of room for both sides to be working.
And I'll just add that, having seen biology evolve so much, there's more tools in your tool set, right? You have access to many more tools. And one thing I think is really important is, if you're doing biology, I think you also need to be more computationally open to computational tools and learning that side of technology, because I think that the amount of data, the systems biology, no one can really interpret it just on their own. You do need these tools in machine learning, so it's really important. But I do believe that at the end of the day, you will have a reductionist view of things for drug discovery. At the end of the day, in my world, we're developing drugs for a specific target. So we'll come down to the actual protein, but it's starting large, focusing on the target, and then going back out again to see if the molecule is actually affecting the system biology. So it's kind of an hourglass shape.
Yeah, I think I just want to add, it's sort of a why not both thing. If you've spent tens of thousands of dollars and however many human hours generating a set, like do your single cell RNA-seq, do the expensive and fancy and sort of thing that's going to give you the right answer, but also throw in, again, something like cell painting that's inexpensive, or do something that's simple that then maybe will help you learn what that more harder to scale assay was later. There's some interesting papers I can point people at where, again, 20% of the time it works 100% of the time, but if you can develop these predictive models, then the next time you can single cell RNA-seq only the exact right sample.
Hi. So first I'd like to say thank you to the panel. This was very informative. This isn't a question, it's more or less a comment. First, Tim, I want to say to you, public servant, to retired public servant, thank you for your service. We know that even though your titles may change, the oath doesn't, so thank you. Daniel P., I was waiting for the word, and you said it, which was humanizing, going from, the lab to the actual person, the actual human. I just want to share that I had the opportunity to participate in the Patient-Focused Drug Development Council. And by that opportunity, it helped me understand the complexity of, the drug development process. But what I just want to add is, again, I think we get the most insight from the actual people or the actual person that's living with the disease. I've been a beneficiary of science and research, but nothing compares to that real life experience of the person. And I just wanted to say that. Thank you all.
Can I add just a quick comment onto that? So I really appreciate that perspective. And I don't do this work personally, but I think what's incredibly striking in the field of stem cell technologies is that ability to, now that we have such robust systems, we can personalize to each patient in the clinic. There are folks that run a clinic and a research laboratory who have that resources and ability to see a patient, retrieve, with their consent, retrieve their stem cells, culture them, and potentially even test drugs or in a cultured dish to see if that could be predictive of if the patient is going to respond to that. So it's really taking, starting from that patient, going into culture, and not just saying generalizing to the population, because there is no generalizable population. It's very specific for each person. So yeah, I think it's a great path forward too.
Thank you to the panelists for great discussion. I want to get back to the point some of you made about how we are all, including us at the Allen Institute, making large data available. So our experience has been you can make data available a ton, put it out there and they will come. It doesn't always work. And if I put myself back to being in the lab, it's just overwhelming. You don't know where to start. Nobody's there to guide you. So I'm wondering, this is also a comment whether some of us can come together, and we've tried to do a hackathon or a workshop. Beth, you might have done the same at the Broad. But is there a way to get people to use this type of data? Because just telling them it's available, I don't think gets us very far.
Yeah, the word metadata was brought up before, and I think, Katerina Strimbrio-Castillo at UMass, I think puts it particularly well that, again, the metadata is the treasure map to where the treasure is buried, and the treasure is your actual data. The problem being nobody agrees on what's the right metadata to capture and stuff, and there's a lot of stakeholders who are now really trying to say, can we get more value-added repositories? So let's not just dump stuff on the internet, but actually dump stuff on the internet with some, things about what that is. And there's going to be a big stakeholder meeting this fall. But if you're interested in that, I would say, especially on the microscopy side, Bioimaging North America, as well as QuaRepLimi, are two organizations that care a lot about this. And I think they will come if we don't just give them terabytes of unorganized data, but a little bit more organization will help.
I also just want to add on to this. We have some documentation that goes along with the data set that makes the use relatively easy, I would say. But we also have exactly those plans. What you just said in a past release, which was a much smaller release a couple of years ago, we organized hackathons around that. And obviously this is, like I said, a cross-fertilization aspect. We got a lot out of this hackathon, a lot of new ideas that we didn't have before, and how to leverage, the value of the information that is in these data sets. So there will be more, and maybe we can connect later and exchange names. So.
Just to add on that, just very quickly, we're taking a very similar approach at CARD. We have our specific data science team that, from the ground up, the goal is everything shared, pre-published, and not just dumping it on a website and saying, here, download my files. But actually, our data science team one of their goals is to make very visually, easily accessible portals where people can go in and look at aggregated data sets from all these different research groups, research organizations, and be able to start comparing those studies even just in that data portal. So you're not just taking someone's FASTQ files or whatever it is and starting from scratch. It has to be very accessible, or else no one's going to look at it.
So
Sorry. So one quick question is, we can really appreciate the power that this technology can achieve. I'm just curious about how proactively you try to be inclusive for ethnicities and you know, like to build up therapies available for like more people, and in line of the panel we got yesterday.
Massively so, like to the point where it's a question again of availability. Like, you know, some of the work that we're doing in sort of that rare disease is quite challenging, because age is also something. I pointed out, age is something that can be quite a big confounder in looking at things. But if you only have you know, research material that's from an affected child who's 2 or 3 years old, and that's fine, that they've been consented in, and you know, that has gone through the right paths. There isn't age equivalent healthy tissue available. No one in their right mind is really collecting punch biopsies from a 2 or 3-year-old. There isn't a need for it, and so it becomes very hard at that level to like make sure that the data is reflective of the age of the individual that you're working on. And then on top of that, ethnicity, yeah, you know, that becomes as much as we can. This is why having the tools that we do, we're very fortunate to have the tools where we can run these experiments so that they can be very big and become more inclusive as you have access to the material. I think it's just that. It's just being in a position to be able to run those experiments of that size. We'll try and make these things as ethnically diverse and balanced across cohorts. And it's just, yeah, at any given time, what is available for us to be able to be used.
Hi, wonderful talk and really really nice panel here. You know, I love learning about the capabilities that NYSCF has, and my question is, this is something I'm exploring in my own research. We are using iPSCs now for drug screening. How about biomarker discovery? Is that an active area of interest, and where does it stand currently?
Yeah, I think it's like right in line with everything else. I think pulling out the phenotypes that become then those biomarkers, or a target.
For translational work,
right?
Yeah.
Sorry.
For translational work.
Yeah, exactly. It's if you can find something that appears to be a biomarker in an in vitro model. Absolutely, can that be translated somehow to be something that could be used diagnostically, potentially? I think we've really focused on this panel of like targets that would be therapeutically relevant to modulate, but it goes very much the other way. I think it's just as applicable to biomarkers.
And if I could just add on a little bit to that for the translational side, you know, as we're getting more access to biobanks and genetic information and uncovering genetic variants that are linked to disease, and we're, as a drug developer, we do want to develop drugs for rare diseases, but as the economy is that we can't make it super rare, so we try to develop drugs that will be beneficial for a wider swath for that disease, for including more patients. And you can't do CRISPR animal models for every mutation that we find from, you know, in particular gene variants. There are multiple. There's one ion channel that I'm interested in right now, and there are multiple mutations, and the children are born with different types of mutations. So having access to the patient samples where we can test our drugs in these stem cells or in these neurons that incorporate real-world mutations to see if it's going to be effective, we would never be able to do that in an animal model. And so that's where I'm hoping the translation will really support more targeted drugs for patients in the clinic. Thank you so much for the panel, and thank you for the question.